Right, we're changing tack now. We're on to the Flash Talk poster adverts, and we only have two because Rachel Dempsey unfortunately has had to drop out. So first up, we have Hannah Goldswain, who I guess doesn't want to hear any more questions from me.
No, thank you, if that's okay. Hi, I'm going to be talking to you about part of my PhD project and a continuing project that's investigating the evolution of seasonal human coronaviruses and their application to future evolutionary directions of SARS-CoV-2. So this is a big project with our collaborators in Slovenia at the University of Ljubljana, who have quite a large biobank of samples. So they have 1,355 seasonal coronavirus samples that were swab samples collected from hospitalized patients in Slovenia and then retrospectively analyzed for seasonal HCOV presence. So this graph, can you see my cursor? Yes. This graph here shows that there weren't equal amounts of samples throughout all the years, but the years cover from 2012 to 2022. And what we decided to do is generate bespoke primer schemes that were either 400 or 1,000 base pairs amplicons generated and sequence these samples via nanopore or Illumina sequencing. And we got pretty good coverage for both types of sequencing. We had a few issues with sequencing the HKU1 seasonal coronavirus, where we got like some of the last third of the genome dropping out. But Anya and the team in Slovenia have solved that problem. And we think that might be due to the fact that there's different genotypes present in HKU1 across the samples throughout the 10 years and still circulating. So the first thing we wanted to look at was characterizing how the genome's changing through insertions, deletions, and SNPs. One of the main takeaways here is that insertions and deletions are kind of static across the 10 years. They don't really change as much, especially in the minor variation, or there's a bit more fluctuation there. When we looked at single nucleotide polymorphisms, we found that in general they increase over the 10-year period. And in dark green here, we've got HKU1, which fluctuates a lot more, which again, we're putting down to there being 3 different genotypes. What this doesn't show or take into consideration is recombination, of which the analysis is ongoing. In order to compare it all to SARS-CoV-2 data, I repeated the analysis with sequences downloaded from GizAid, which you can see on here as well. SARS-CoV-2 is increasing slightly more in the SNPs over time, which you might expect because it's a novel virus, still getting used to its niche. Also completed a phylogenetic analysis, and sort of the immediate takeaway from this is that all of the viruses look different, slightly different. Longer branch lengths could be indicative, well, they are indicative of large changes from previous samples, which could also represent recombination. If we look here at HKU1, and this analysis is currently being completed by Kate at the University of Edinburgh in Andrew Rambell's lab to fully unpick how much recombination is contributing to evolution over time. I'm going to skip my conclusion slide and say that you can find out more at my poster, which is poster number 4. And thank you to our collaborators in Ljubljana for all their hard work and the Hiscox lab. Thank you.